Welcome to a whirlwind tour through the tiny bustling world inside our heads, where molecular neuroscience meets imagination. Suddenly everything that's seen invisible gets a vivid, almost cinematic look. At the heart of it all is the nanoscale organization of synapses, the precise dance floor where proteins like PSD95, neuroligins, and NMDA receptors are forming a nano-architecture that shapes synaptic strength and plasticity. Imagine mapping receptor distribution with DNA paint, pushing resolution beyond the diffraction limit to about 5 nanometers, so we can actually see how molecules rearrange as signals pass from one neuron to the next. And STED, microscopy, and SMFRET-based sensors, and we're not just watching static pictures, we're visualizing conformational changes in glutamate receptors under stress or drug action, watching the choreography in real time. In this nano realm, not only do we observe structures, we witness function, nanocolumns aligning pre- and postsynaptic elements to optimize neurotransmission, a phenomenon that starts to reveal how precise nanoscopic spacing might influence a whole brain's behavior. And because the nano matters, researchers are asking big questions. How does nanoscale organization relate to psychiatric symptoms? How might aberrant nanocolumn spacing connect to schizophrenia models? Or reduced AVPA receptor mobility linked to depression like phenotypes? The answers are not just academic, they hint at restoration strategies. Ketamine or SSRI treatments, for example, can reset or retune nano-architecture, suggesting nanoscale reorganization as a potential biomarker for precision psychiatry. To keep pace with this rapid frontier, scientists are embracing DNA mechanobiology and the world of motor proteins, myosin, kinesin, domain, along with the cytoskeletal tracks they ride, actin filaments, and microtubules. These nanoscale biophysics are not just scaffolds, they're movers in a dynamic system that translate mechanical cues into functional states. Imagine a landscape where nanotechnology, motor proteins, and mechanosensors work in concert to illuminate how synapses remodel themselves in health and in disease, guiding us towards nanoscale functionality, connectomics, and targeted drug screening at single molecule resolution. In this view, a biopsy might become a nano-imaging expedition, charting how synaptic nanoarchitecture shifts in different psychiatric pathologies, and how iSPC models can recapitulate these patterns in a dish. So it's a playful, high-stakes quest to translate the tiniest of scales into meaningful, measurable improvements in mental health, and maybe even reveal the hidden logic of consciousness itself as it plays out across neural networks and synaptic nano landscapes.